Hello again. Okay, I'm still at the tutorials, and this time we are going to do this questions from this tutorial, 2018. All right, here it is. Okay, I'll just enlarge it a bit. The first question, this is one of the example of exam question. It says carnation and garnet are two X-linked mutations that can produce brown eyes in Drosophila. Design an experiment to determine if the two mutations are allelic. This is the complementation test. Basically, what experiment you want to do is to try to get heterozygotes. You do crosses, and then these double heterozygotes can either be in coupling or in repulsion. Remember whether it's cis or trans configuration. You look at the phenotype in which the cis configuration serves as a the cis heterozygotes they serve as control, in which both if they are allelic, that means they are one gene, it will show wild type. And then if they are non-allelic, that means they are on different genes, it will also show wild type. The defining experiment would be looking at the trans heterozygote, in which if they are a single gene, if they are trans heterozygote, that means both the copies will not be functioning, so that means you'll get a mutant phenotype. Whereas if it's they're non-allelic, that means they're different genes, then what you will get is the wild type phenotype. Okay, so focus on the trans heterozygote, in which if they are from single gene, the trans heterozygotes, that means they are single gene, means they will have a mutant phenotype. If it is non-allelic, then they will have a wild type phenotype. Okay, now so that answers that question. We shall move on to the next question. This is on epistasis. I'll just adjust it a bit. Okay, here you go. Hall and Aldous found that a white fruited form of blueberry, I'm reading the English question, Vaccinium augustifolium, which they crossed to a true breeding blue fruited variety. So you have a white fruited blueberry, then you cross to a true breed blue fruited variety. Then of course you get the F1. The F1 offsprings of this cross were 30 blue fruited plants. So that gives you an indication that the blue gene or the blue phenotype is actually dominant. When the F1s were crossed with each other, and then you have 127 blue fruited, you get 127 blue fruited, and then you do get 4 white fruited plants were produced. Now again, when you're doing epistasis, so you need to talk about numbers. Okay, and before we go further, again I will do this. 1, 2, 3. There you go. And then I will change the view so that you can actually watch me do the working. Okay. So 127 and 4. So what you have after the F1 times F1 cross, you get 127 blue and then 4 white. Remember, in the P1 cross, it is white versus blue. Okay, I'm assuming. Okay, never mind. We'll work on that. Now looking at these figures 1, 2, 7, and 4. I am inclined to believe that this is the duplicate dominant gene that gives you 15:1 ratio. Now if it's 15:1, so let's just add this together. This will be 131, and then if you divide this by 16, you will get about 8. Okay, now and 8. So if under the ratio of 15:1, 8 times 15 is, what is 8 times 15? 8 times 15 is 40. 8 times 1, 8, 12, 120. Is that correct? 5 times 8 is 40. 8 times 1, 8 is 120. So this is, you're expecting 120, and then and then 8 or 7. Yeah. Right, so this is, oh, because this is 8 point something something something. All right, okay, never mind. So you are let's just use the calculator. Can you see my calculator? Okay, so 131 divided by 16 is 1875 times 15. There you go, it's 122,123. Alright, so this is 8.1. Okay, so it's about 1.123. So then you have this will be your expected number, and then this will be your observed, and then you can do a chi-square test, right? So how does this work? 15:1. Let's look at the slides. There you go. I've actually looked at this. So wait, before we do that. So duplicate dominant genes, 15:1. So you have here A and B is same expressions. That means either you have big A, either you have big B, you'll get the same expression. Alright, so A equals to B, right? In terms of expression, right? So A is dominant to A, B is dominant to B. Okay. A and B same expression, which means to say that it doesn't matter what you get, as long as you have either A or B, you'll get the same phenotype. And then only in conditions where you have A, B, B, you'll get the other phenotype. I'll call it the second phenotype. So in this cross, Punnett square, AB, AB, and AB, AB. So as long as you have A, you'll get the first phenotype. So right, so this one has got A also, this one has got A also, this one has got B, this one has got B, and only this one has got the other phenotype. Okay, so what's the question? What is the hypothesis you were offered to explain for this result? This is duplicate dominant genes, and duplicate means you have A and B same expression. What experiment lanjut? What further experiment would you undertake to test your hypothesis? Well, if you know what this is, then basically, if you know that this is the situation, so what you need to do is maybe you can do back cross or whatever cross you can do, and then as long as you can predict the outcome, then it should be fine. Okay. All right. I think the most of the marks will actually be in your workings. All right. So I'll see you in the next question. Bye bye.